Now we are going to talk about the real-time monitoring, uh, inline and online sensor. What is a real-time monitoring and inline and online sensor? Uh, as we discussed in earlier lectures where we talk about the bioprocessing of the different parameters, for example, we talk about the temperature, we talk about the pressure, we talk about the pH, we talk about the dissolved oxygens and gaseous interchange, nutrients, conditions, right? We talk about the PID system, right? We talk about the SCADA, we talk about the data analysis. Now, what data analysis is being made in order to obtain the best quality product? It could be of manually, which is very, very difficult. Now, in order to maintain them, to obtain these conditions, or to rather attain these conditions. So before that, we simulate and we make a different batches, and these batches show the quality, quantity of the product, bioprocess product, which give you an idea what are the conditions required are used in order to attain these batches of the product. So how do you understand, how do you know the variation in these parameters to be changed, to be regulate, as we just talked in earlier lectures, regulation of this bioprocessing, right? So this is how the real-time monitoring is important. Real-time monitoring means like during the process of bioprocessing, during the whole course of bioprocessing, say for example, 2 hours to 24 hours to few days, for example, depending upon the cell line, cell cultures, or bioprocess product you are obtaining. You have to, at every point of time, you can look into the product development in the form of like peaks. And this real-time monitoring will give you the best quality product and economically good. You can control the nutrients, you can control the conditions as and when required. So it may be inline and online sensors. It depends on the machine system. So real-time monitoring, inline and online sensor means like inline monitoring means like a sensor placed directly within the bioreactors. So this is how a different kind of a sensor, as we discussed in my earlier talk, within the bioreactors provide the continuous real-time data without sample removal. So you need not to remove the sample to check it through spectrophotometry or through some optical sensor or through some other kind of a sensor to check whatever you want. No, it is within the sensor is there, and this internal sensor or inline sensors can give you the idea that these systems offer the immediate feedback based on the data you obtain for the process control and enabling the rapid responses to changing the condition. And this signal will give you, okay, the things are changing and the outcome of the product will be not good, so you keep change the conditions. So what are the condition to be changed? That you have to interpret, that you have to look into from the PID and the data analysis on the long-term data analysis that you have done, and the data is available with you. Now the online monitoring means like you have online sensors, the automated system that analyze the diverted sample continuously and providing the near real-time analysis of a critical parameters, right? And this approach combines the benefits of a real-time monitoring with analytical precision. Now, why it is required? Because in this case, you will get very precise changes or precise modifications which are being made through these through this analytical process, through this sensor process, and these both approaches enable the immediate correctiveness, immediate change and modification, which can reduce the risk of process in terms of like yield, in terms of like a quality, as well as in terms of like a safety parameters. Offline and at-line monitoring is called sample collection. Sample collection, sample taken at predetermined interval, for example, intermittent collection of a sample from the bioreactor at different batches or within the batch at different time period, uh, using the sterile condition, of course, to maintain the process integrity. And this data integration, like results combined with real-time data for comprehensive process, understanding and decision making will be done. And laboratory analysis means like a comprehensive testing for cell viability, whether cells are viable or not. Metabolites concentration, what is the concentration of metabolites? accumulating during the processing, and product tighter and contaminants. So all these things can be offline monitoring will be provided, and the manpower is required to obtain all these things. Right. So the automated sample system reduce the manual errors and increase the sampling frequency, providing the more detailed process insight. And these system uh, complement real-time monitoring by offering the detailed analytical information not available for inline sensor. Okay, now the modern control algorithm. What is the modern control algorithm? To understand the modern control algorithm is it is the algorithm which is based on certain tools, are AI based or machine learning tools. Now these machine learning tools and AI based tools provide some algorithm, and these algorithm will be like robotic system will be designed in the system or in the machine or in bioreactors which is connected with the system RUP, we call the CPU, with some high power system where the algorithm is being run in the program and the software control the whole monitoring. So we call proportional integral PI control. It provides the stable set point maintenance by combining the immediate response to deviate with the corrections of a steady state. Errors form the foundations of the most bioprocess control process. Then we have adaptive control system where dynamically adjust the control parameters, right? Then we have AI machine learning. In AI machine learning, the advanced algorithm which predict the optimal conditions, so they can have design based on what is requirement, both in quantity and quality, and what are the parameters through simulation are required to be given in the algorithm, and accordingly this algorithm monitor if it's slight deviation taking place in the parameters. So it will give you the alarm, it will give you the signal, and that signal give you okay, this is how you can maintain the whole process of uh, regulation so that you can get the best quality product. What are the new trends and emerging trends on in order to uh, provide the best quality product in bioprocessing and bioreactors? The cloud-based platforms, uh, remote monitoring and control system is also using the cloud base. It is a global process, right? Oversight and data sharing process across multiple facilities. Then PAT integration. This is called process analytical technology framework combine multiple sensors for comprehensive real-time process understanding. Now, advanced AI system is a neural network and fuzzy logic, handle complex multiple parameters, optimization in sophisticated bioprocessing application, and these emerging technology promising, promise to revolutionize the bioprocess control in the bioreactors. They may also enable the more precise, efficient, and intelligent manufacturing system for biotechnology industry. So having these emerging trends in the regulation system, in the process of fermentation, in the process of the bioreactor and the bioprocess product, it is important that to have these new technologies which are either AI based technology or PAT integration technology or cloud based technology which can remotely control the whole process of integrations of these bioprocess product. Now the advanced control strategies, the modern predictive control we call MPC. Well, it is a it uses a dynamic process models and predict the future behavior. It means like you can have a predictive model. Again, this is AI based model where you can predict the possibility of like failure or the possibility of like quality improvement system. So that could have predictive models can be generated, and predictive model based AI predictive model based controlled system are being inbuilt in this process, and behavior of the cultures can be identified at the optimum conditions. The ideal for complex multivariable bioprocesses to optimize feed rates, temperatures, and dissolve oxygen level for maximum yield. Now, of course, with this advanced control strategies, we can produce the maximum yield. Fuzzy logic control is another very interesting area. Fuzzy logic is implies if and then. So there's a, always there's a confusion. Yes or no, yes or no. So you have to have like rule based on expert knowledge and linguistic variable, mimicking human decision making, like effectively for highly non-linear or ill-defined bioprocesses where the precise mathematical models are generated, are developed, and which are difficult to develop, but you can use these physiologic process in order to overcome these models if you cannot develop these mathematical models, such as the controlling cell growth phase. Which phase of the cell growth are to be maintained can be looked into this method, physiologic. This required basically a lot of algorithm and computer knowledge to in order to maintain the machine. So there are machines which have now developed, are manufacturers having this information about the using the fuzzy logic process. Then another AI tool is neural network. This is again algorithm. In AI, neural network control is like ANN, artificial neural networking is one of the algorithm. It utilizes the artificial neural network to learn intricate process. Now, these intricate processes dynamic for historical data. It provides a robust adaptive control system to optimizations. Now, again, these systems are very robust and they can optimize the growth conditions, all variable factors like a temperature, pH, pressure, and nutrient conditions, dissolve oxygen for the good quality bioprocessed product. Uh, Fault detection. So what are the different faults may occur during the process, right? It could be machine fault, it could be nutrient fault, it could like anything. It could also be having fault which could have like risk to the operational manpower, operating manpower for the safety point of view. And it can predict the process behavior, it can predict the complex biomanufacturing, the environmental conditions in order to in order to maintain the deviation of these from the higher side to the lower side. Adaptive control system is automatically adjustable system where you can control the parameters in the real time to compensate for the change in process dynamics for external distribution, ensure stable and optimal operation even when process conditions are less characteristic evolve or over time improving the consistency. So these methods, like including the MPP, MPC, fuzzy logic, neural network control, and then adaptive control system, are the system which are modern system to maintain the biofermenters and bioreactors in order to maintain the all physical parameters and variables to maintain at the optimum point to provide the best quality bioprocess product. So this is the purpose of this. Now, sterilization and contamination control is again important because why sterilization and contamination control is important, because if the machines are not maintained to maintain the sterilization conditions as we discussed in the topic of pressure, immediate sterilization or sterility maintenance, then contaminants may come across and contaminants may mix with these cells. For example, if you are working on a mammalian cell line for some product development, and these mammalian cell lines may have contamination with the bacteria, right? The it will directly affect the yield. It will compete with the growth of the mammalian cell, or it will damage or destroy the mammalian cell, or the quality of product will be affected. Similarly, if you talk about the wine production for wine production using the Saccharomyces cerevisiae or baker's yeast. Now, the quality of baker's yeast get contaminated with the bacterial population or some strain which is not of good quality. Then it compete with the good quality and bad quality strain together, and that way it again impact the yield of the bioprocess product. So in order to get the high and good quality product, good yield of the product, bioprocess product, the sterility to maintenance is very important to avoid the contamination. Therefore, contamination control to be on the priority basis. Now, in order to maintain the sterile environment, it is important that to bioprocess prevent contamination process and maintain the sterility. So steam sterilization is the best sterilizations before you process your product. So everything should be steam using steam sterilization. So utilize autoclaves to eliminate the microorganisms, are especially undesirable microorganisms, right? From the equipment, glasswares, culture tubes, media, and high pressure saturated steams can use to sterilize all these products under certain experimental condition or for certain time period. Now, the forming the first line of defense, that is to control these contamination, is important to use the steam sterilization. Now, filtration system is again, it employs the specialized membrane filter of the different growth, control the growth, undesirable growth. These filters are made based on certain, the membranes, and these membranes are of some pore size, and the pore size are such that they can avoid the uh, filtration through of the microorganism. They can rather retain the microorganisms or microbial flora, which is not desirable from the media, from some other aseptic filtrations, liquid media, gaseous airs like O2 and CO2 dissolution, and it also removes the uh, bacterial viruses and other things uh, for as a contaminant to be added in the product. Now, if the filtration is it is a second line of filtration where things are remain sometime there in the media or in the glasswares which are being used, or the vessels which are being used, then through filtration process, this liquid and gaseous can remove these contaminants through the filter, using the filter. Now, the aseptic techniques, strict operational practices, where there should not be, aseptic means like no septic or no kind of infections through non-desirable or contaminant bacterial populations or the fungal populations. Even if you are working on the bacteria for production, the other bacteria may not be mixed together. For example, if you are focusing on E. coli recombinant protein productions like insulin, and the E. coli get mixed with the other E. coli or with some Klebsiella pneumoniae or some other species, it compete with the nutrient and the yield will be reduced. Therefore, the aseptic conditions are very important. So you should have a controlled, clean room environment, then minimize the introduction. So wherever you are working for after the bioprocess product being canned or being like tinned food, like the various aluminum films are used to keep these pharmaceutical products or whatever. So during packaging also, it is important to maintain the aseptic conditions. Otherwise, if you packed these sterile product and during packaging, if you got contaminated, then that could have created the problem from the human safety or the patient safety, or the if it is biopharma product. The microbial monitoring is again continuous monitoring process. So sometimes real-time monitoring process is also being done. Uh, so bioprocess product in environment in which it is being produced, what is the raw materials? How purified the raw material is, right? What you're expecting from the raw materials to produce, So all these parameters should be taken into consideration by through monitoring these separate conditions. Now, the cell viability and growth monitoring. Uh, Cell viability is important because if cells are not viable or not maintained to be viable, then of course the processed product will not be of the required yield. So for successful bioprocess development, manufacturing ensuring optimal conditions for cell cultures and maximizing the product yield process. So cell counting method. So again, the saturation of a cell, the cell should not be dead. Cell should remain maintained. So for that, you need to have the manual hemocytometers and automated system quantifying the total cell density. Now here, one thing I would like to share, that if you talk about the hemocytometers, are some system where you can microscopic method, or for that matter spectrophotometry method. That way it will not give you the idea about the live and dead cell, unfortunately. Therefore, you need to have the intermittent during the culture. After a certain time, when you are supposed to have the loss of viability, you take the culture under the sterile conditions and go for the CFU, colony forming unit, in the viable cells, and see, are the cell growth is improving, or reduce, or like cells are dying. So that can be maintained through this. But this is a very cumbersome job, because every time at every hour and every 2 hours you have to take the cultures and dilute it and grow again for 20, because it takes 24 hours to give you the results, or it give you like 16 hours. So by the time you have already crossed the batch to get the product. Therefore, what the people do, they have a cell density plot. So they can have a, they know if at this density of cell, what is the number of viable cells might be there. So that can be controlled through cell density, essential for tracking the population size, that can be the growth phases. And if it is remain in that cell density range, the growth phase in the log or exponential phase, you can save that, have all the 100% cells are viable. So you need not to worry about it. If it crossing that barrier, if sorry, if it crossing to that density, and it might give you the indication that it might reach to the stationary phase, then probably you need to have worry about it, and you have to do some exercise in order to either provide the nutrients or exchange the materials, byproducts, and you can process the byproduct at that point of time. Viability assays, as I've just talked about the different viability assays. So techniques like we have a trypan blue exclusions and fluorescence dye, which differentiate between the live and dead cells, providing the critical insight into the cultures and stuff. So these things can be done easily. Now, growth kinetics is another things, and the real-time monitoring. No, but in the growth kinetics, you need to have real-time data required for doubling the times, and you can have number of the cells increase in a minimum quantity of minimum amount of time. So you can measure the how fast the growth rate is, and if the growth rate is get reduced after a certain time, then you should provide some nutrients or regulate some conditions in order to provide the best quality cell culture. So real-time monitoring is important. Inline sensors which can give you the indication the cells are going down and growth rate get reduced. So you can have like different optical and capacitance probes can be used to provide the continuous non-invasive data. Offline data, besides offline data, online data analysis can be done through these sensors. And this monitoring technique system will provide you the best possible information to give you the regulated bioprocess product. Now the metabolite analysis and product monitoring system, uh, yeah, done by HPLC is high performance liquid chromatography. Then you have a mass spectroscopy. So precise measurement of these metabolites or the bioproducts can be done by HPLC, high performance liquid chromatography. So it separate identified the quantification of a product, a single product, purified product based on the specific column which are being used, right? So this is how you purify, how you biopharmaceutical products can be purified and can be detected, can be identified, and the concentrations can be identified through these high machines. Mass spectroscopy provide the highly sensitive and specific identification of the quantitative product. So you know the product being obtained, right? You know what wavelength is required in order to excite that wavelength to measure under the spectroscopy can be identified, and the concentration can be calculated. So that concentration gives you an idea of what is the total amount of product is being prepared, is being manufactured in the given volume. Uh, the enzymatic assays and real-time product quantifications again utilizes the high specificity of enzymes to rapidly, accurately quantifying key metabolites, substrates for the products like galactose, lactose, amino acids, and different cell product. Could be primary products and primary metabolites. In integrate time product quantification, you have integrates in line and at-line, inline and at-line spectroscopic techniques, and uh, near infrared Raman spectroscopy for continuous non-invasive monitoring system for getting the product concentrations and enabling the dynamic process control. Now, these techniques are utilized for in the real-time manners in order to control this product byproduct through these biofermenters and bioreactors. The case study AI-driven temperature agitation control machine learning process model is used to successfully predictions of optimum agitation process, for example. Now the speeds for the yeast fermentation process, for example. So 50% ethanol yield increase. It has been observed in one of the case study uh, through driven by AI, both temperature and agitation control. So if you control the temperature and agitation in order to regulate the bioprocess product, 15% yield can be increased in ethanol, 10% energy reduced, 10% energy reduced to get it. So low energy, low power consumptions, through intelligence agitation process. So this is one of the successful model where you can use AI tools to regulate the temperatures, to regulate the agitation process, to increase the yield, reduce the requirement or consumption of the energy. This is how wonderful the system is. Challenges in bioprocess monitoring like sensor foulings and drift, the intracellular measurements, then we have data integration complexity. So these are some challenges which these challenges drive ongoing research. So you can improve these challenges and you can control the bioprocess monitoring, crossing these challenges. And because monitoring internal cellular metabolites remain technically challenging process, then combining multiple parameters data is streamlined into the action of like AI based tools and other is required a lot of sophistication. So to detect, so manpowers and skilled people are required. So every time you may not have that. So this is again a big challenge to get the manpower to understand the whole process of integrations and getting the data. Now, in Indian biotech context, the growing biopharma sectors, automation adoptions, government support, like India's expanding the biopharmaceutical industries, demanding the robust bioprocess product development. Then increasing the implementation of the automation system, smart sensors, optical sensors, new technology, AI-based technology. So this is all new prospects of the Indian biotech companies. Government is supporting national initiative support like biotechnology, startups, innovations, quality standards, and create a favorable conditions for the youngsters to move on for the startups process. So Indian biotech sectors position for the significant growth with government and support, the increasing the investment and growth expertise and all. The the this is how toward smarter, more efficient bioprocess product by master the fundamentals, embrace technology, future innovation. So now, in order to be more smart and get the efficient bioprocess product, we are in future product development, in biotechnology product development, pharma product development. We need to be embrace the technology. We need to adopt the technology, and we look into the more future innovations in modification of this technology, how this technology can be further modified to get more better quality product. Now, the bioprocesses safety, again, the proactive safety measurements, comprehensive risk assessment should be taken into consideration. Emergency responses planning is important. So, ensuring the safety and personnel, both operating system, those who are operators for the systems, is very important, because if the biosafety is not being attained, then we may have at a risk to get these byproduct put at the toxin productions, hazardous product development that have high risk factors toward the patient's safety, patient's biosafety, and also the those who are involved in the manpower which is involved in the process development, so reactor operation, right? All these factors are important to maintain the safety. So safety protocols are very important. SOPs to be maintained, both to run the bioreactors and to operate the bioreactors also, right? To provide the raw materials and what raw materials to be required, everything has to be properly under the SOP condition. Bioprocess scale-up challenges, equipment scaling, this is one challenge. The process optimization is another challenge. The quality and regulatory control is so we have already discussed these process optimization, which is one of the scale-up challenge, where at industrial level, at the laboratory and pilot scale, we can do it. But I think sometime when we go for the liters and gallons of the product development, then it is a big challenge for the operators to operate such machines, maintaining the safety of its own and the product safety. Regulatory compliances in bioprocessing. Key regulation is documentation requirement, role of quality assurance, like again based on the operation systems and based on other parameters, both in the variables like temperature, pressures, and other things. Some key regulations are required, like we need to have like FDA approval things, and several regulatory bodies are required to approve the bioproducts for the use of the as a trials, as human trials are phase 1, 2, 3, 4 trials, or if it is already trial, then again we have to have approved these products which are being made through this processing. Sustainability in bioprocessing, green chemistry, waste reduction, and then we can have the energy efficiency. So how to sustain these? So it is important that to maintain the raw materials, which can have high sustainability for the bioproduct processing through green chemistry, through the utilization of the waste generation, optimization of water usage, and exploring bioproduct in order to produce the bioprocess product required for the various pharma industries and food products, focusing on energy efficient equipment so that we can have a best quality equipment. So energy can be reduced and product can be increased. The bioprocess innovation future directions like emerging technologies, key research trends, and future landscapes. So we should look into the new technologies, innovation into the new technology, both at the instrument level as well as in the the biological sample level. For example, the strains are in the bacterial population which are used are different strains to be modified, genetically modified strains to be used for high yield, increasing high yield producing strains. Now, we conclude that the future bioprocess control monitoring is basically uh, is important that optimize the process development, that precise control, optimization process parameters, advanced technologies, integration, then future forwarding processing. So we should have a precise control processing variable, temperature, pH, pressure, dissolved oxygens, and other yield, ensuring the consistent quality in bioprocess product. Then advanced technologies to be integrated, right? And then future forward bioprocessing technologies through AI based and machine learning based technologies should be incorporated into the bioprocess product. So these things are important for best quality bioprocess product. With this, I thank you very much for listening, and I think we have some more questions in the next session. Thank you so much.